are thrilled today to present our study on manganese mineralization on bacterial surfaces as part of the prototype flash episodes. And our project was actually initiated when I was a PhD student at Institut de Physique du Globe de Paris. And at that time, I was studying how microorganisms could control the immobilization, transport, speciation, and eventually toxicity of dissolved metallic ions by inducing the formation of minerals in the environment. So to bring a little bit of context, on the surface of our planet, microorganisms, which represent the most important fraction of total carbon mass in the environment after plants, are found in the form of more or less thick microbial mats covering the surfaces. And at the microscopic scale, the biofilms correspond to a porous network of cells connected to one another by organic polymer that they secrete. They are called the exopolymeric substances, or EPS. So when a biofilm is exposed to a solution containing dissolved metallic ions, the porosity and the negatively charged chemical functions at the surface of the organic matrix are like affecting the concentration of precursors in solution and therefore the supersaturation with respect to a mineral phase, and they can induce the nucleation and growth of a mineral. So many studies has observed this phenomenon at the submicrometric scale, but there are very few reported the process at the spatial scale in hydrated conditions, avoiding therefore the shrinking of the fragile 3D structure of the organic matrix. So the goal of this work is therefore to understand the role of the hydrated organic matrix in the sequestration of metallic cations at the submicrometric scale. And this is why we contacted Damien at the MPQ.
Prior to our work, many groups have already studied the mineralization process by liquid cell TEM. But so far, the nucleation and growth of minerals were only studied directly in solution or in biomimetic media containing specific polymers. But on the other hand, liquid cell TEM has also been deeply exploited to study the structure of biological spaces directly in their native condition, including bacteria, viruses, proteins, and extracellular vesicles. So the idea of our work, the challenge we try to meet, is to combine these two know-how to observe the mineralization process directly in biological environment and use the bacteria as a template to reveal the effect of bacterial surface chemistry on the nucleation and growth process of manganese. And to do so, we exploit the Poseidon select order from polar chips, which allows to study dynamical process at the nanoscale in liquid environment of well-controlled composition. And we used the flow spacer liquid cells with a flow spacer of only 150 nanometer gold spacers in thickness, which was the best compromise to be able to observe micro and large bacteria with a very good signal-to-noise ratio.
So as mentioned previously, the goal of his work is to understand the role of the hydrated bacterial surface and exopolymers in the sequestration of dissolved metallic cations, and therefore to better understand their influence of mineral nucleation, nucleation and growth. We exposed in manganese-bearing solutions 2 strains of Escherichia coli that differ in the nature of the exopolymer they secrete. So the first strain, Escherichia coli 2K7 in red, produces an abundant and dense cellulosic exopolymer. And the second one, Escherichia coli MGF in blue, this one, on the contrary, does not secrete this bulky exopolymer surrounding the bacterial cell, but produces rather nascent pili on the bacterial cell surface. And the 2 bacterial strains also differ by the abundance of charged chemical functions on their surface. And to illustrate it, the electrophoretic mobility is generally used to measure the overall charge over the surface, is used to characterize this abundance of chemical functions. So here, for example, you see that the electrophoretic mobility of Escherichia coli MGF is about 10 times lower than the one of Escherichia coli 2K7. And so this is due to the fact that some chemical functions on the surface of Escherichia coli MGF are negatively charged at the investigated pH, such as the carboxyl acidic functions and the phosphoryl as well. And on the contrary, Escherichia coli 2K7 only exposed the hydroxyl function of the cellulosic polymer to the solution. And so therefore, this reflects a substantial higher density of negatively charged sites on the surface of Escherichia coli MGF as compared to the Escherichia coli 2K7 and the associated cellulosic exopolymers. And therefore, to compare the role of each bacterial surface in nucleation and mineral growth, bacteria were loaded in a liquid cell with an aqueous solution containing manganese, and in two duplicate-independent experiments, the cells were exposed to the incident electron beam to trigger the mineral formation. And so here is what we observed. In both of the cases, we can see that the contrast of the bacteria increases slightly over time due to the formation of a manganese-rich shell on the surface of the bacteria. The electron beam here is inducing the precipitation of manganese at the surface of the organic template. But the two time series show, however, the difference in mineralization. So, for example, we see that for Escherichia coli MGF, the mineralization is forming homogeneously all over the bacterial surface, but we don't see any mineralization forming on the surrounding medium. While on the contrary, for the example of Escherichia coli 2K7, we observe the formation here of isolated spherical particles that are growing on the cell surface, but also here on the surrounding medium at the vicinity of the bacterial cell. Therefore, this study using a prototype liquid cell made it possible to visualize the manganese mineralization on the hydrated bacteria and its exopolymeric surfaces, and the location and size of the minerals are controlled by the density of charged chemical functions associated with the organic matrix. So, for example, the first strain, to summary, the first strain had a lot of negatively charged chemical function that resulted in numerous nucleation sites for precipitation that further grows in an homogeneous shell over the bacterial surface. On the second example, the smaller abundance of charged chemical functions on the surface of the bacterial cells and the cellulosic exopolymers led to a fewer amount of nucleation sites, and therefore lead to isolated and spherical particles growing over the surface. So these results demonstrate the possibility to monitor the sequestration of dissolved metallic ions in solution in hydrated condition and at the submicrometric scale. And with this, I will let the last words to Damien.
This work and this ongoing collaboration that we have with IPGP perfectly illustrates the interdisciplinary potential of liquid cell TEM, which provides mechanistic information that are crucial for Earth, life, and material sciences.